Dr.K.KAVITHA, Associate Professor, Department of Foods and Nutrition, Vellala College for Women. I am going to deliver about lactic acid bacteria, production and application of lactic acid bacteria. So before going to start the production, we have to know what are lactic acid bacteria. So lactic acid bacteria, these are gram-positive microorganism which catalyze negative microorganism which is used to produce fermented products. And also this is the key organism which is used for the production of yogurt, cheese, sourdough bread and wine fermented products. And this is also helps to provide desired sensory changes, extended shelf life and product safety. And this microorganism which also produce a wide range of organic acids such as lactic acid, acetic acid, citric acid, succinic acid, propionic acid and more. So this is the first organic acid which was produced by microorganism, and nowadays the lactic acid which is produced completely by both the microbiological method and chemical methods. So this is the structure of lactic acid bacteria, and the International Union of Pure and Applied Chemistry name of lactic acid bacteria is 2-hydroxypropionic acid. So this lactic acid which occurs in two isomeric forms like L plus and D minus. So this L plus isomer, it is the most common form which is found in living microorganisms. So this is the history of lactic acid and lactic acid bacteria. So during 1780, Swedish chemist Carl Wilhelm Scheele, who discovered the lactic acid from the sour milk, and during 1857, Louis Pasteur, who is the father of pasteurization, who demonstrated the production of lactic acid by the fermentation process like living microorganisms, and it is a pure natural fermented product. It is not a chemical process. And during later 1880s, so this is the first industrial fermentation process which was introduced for the production of lactic acid, especially from the food industry and leather industry. And during 19th and 20th century, it is defined lactic acid bacteria is a starter culture which are standardized and adopted across the dairy industry and also the other food industries. And currently, lactic acid bacteria which are engineered and then it is optimized for the production of probiotics and novel enzymes and also the functional food development. And this next is classification of lactic acid bacteria. So this lactic acid bacteria which are classified in two groups according to the hexose metabolic pathways. So the first one is homofermentative bacteria which are produced by lactic acid, and these are the primarily that is a byproduct of glucose fermentation, and it can be converted one glucose molecule into 2 lactic acid by the process glycolysis. And the genera like species which are used for the production of this lactic acid bacteria is Streptococcus, Lactococcus, Enterococcus, Pediococcus and Lactobacillus. And the next method is heterofermentative bacteria. So in this method, additionally other byproducts also produced along with the lactic acid such as acetic acid, carbon dioxide and some aromatic compounds. So in this method, the producing lactic acids are it is a mixed form of acids when compared to the homofermentative method. And the genera used for the production of lactic acid bacteria is Leuknostoc, Onococcus, Lactobacillus. So this is the metabolism which are involved in the production of lactic acid bacteria. So the primary product is glucose. From the glucose, it can be converted into the lactic acid. So from glucose, the 2 lactic acid which are produced by the process homolactic metabolism with the presence of enzyme lactate dehydrogenase. And from glucose, there are along with acetic acid and ethanol, lactic acids are produced by the metabolism, hydrolactic metabolism, and with the help of the enzyme alcohol dehydrogenase. So next we can move on to the production of lactic acid bacteria. So before going to that, we have to know the characteristics and growth conditions of the lactic acid bacteria. So the optimum temperature for growing the microbes is 25 to 35 degrees Celsius. For this lactic acid bacteria, 10 to 40 degrees Celsius. And the pH which are required to tolerate the growth range is 3.2 to 9.6. But for the production of lactic acid bacteria, 4 to 8 is moderate. And the mesophilic bacteria which are produced by this temperature and the pH. And some strains maybe grow below 5 degrees Celsius, and also the highest 45 degrees Celsius based on the adaptability. And here the lactic acid bacteria, it is a gram positive and it is a catalase negative and non-spore forming bacteria, and it is also depends on the nutritional, so it can grow this optimum temperature and it can be tolerable for releasing the fermentable, it can be produced fermentation environments and from refrigerated conditions like dairy products to ambient sausage curing. Next is production of lactic acid producing bacteria. There are two methods involved in this. One is chemical synthesis and another one is microbial fermentation. In chemical synthesis, so there is a petrochemical feedbacks which are used for the synthesis of lactic acid through the chemical process, and in this process it is based on completely non-renewable petrochemical sources, and the energy consumption is higher, and it can produce other byproducts, and also the waste is more, and the purification is very complex, and because it requires many technical aspirants, and it is also very the environmental impact is higher. And in microbial fermentation, so this lactic acid, can be produced by natural products, natural substrates like renewable substrates like glucose, whey, and molasses. So this lactic acid, when decomposes these glucose, whey, and molasses, it can be converted into lactic acid by the presence of sugar. And so after when fermentation occurs, the lactic acids are produced. So based on the renewable biological resources, it can be very easy and the energy consumption is very low, and only few byproducts can be produced, and the wastage is very less, and it can be produced easier by downstream processing, and the environment impact is very less. Okay, let's see the production process involved in this. So this is the flow chart. First, we have to select the suitable renewable raw materials, very cheap raw materials, which produce carbon source. So corn, molasses, whey are selected here, and these, after selecting the raw materials, it is involved in the preparation and media formulation. So all the raw materials are mixed together, and we have to adjust the pH here, and the nutrients are added into that media formulation for the media preparation. And after that, sterilize the medium and add the inoculum. So here the sterilization heating process is involved. After sterilization, immediately it is cooled, and that inoculum like lactobacillus lactic acid bacillus strata culture is inoculated. So followed by fermentation is involved. In this fermentation, the pH should be controlled, the temperature and time should be maintained, and the agitation is a continuous process. Here the agitation, so there is a mixture of fermented products, and finally we can get the fermented lactic acid. And after that, so there is a recovery and purification step. In this method, we have to remove the cell debris or any unwanted materials, any impurities can be removed by the filtration process, and the lactic acid solution is purified. So finally, we have to concentrate, and we have to recover the lactic acid by in the form of pure crystalline form, or in the form of powder, or in the form of semi-solid liquids. Okay, next we can move on to the applications. So in this application of lactic acid bacteria, first is starter cultures we have prepared, and we have produced the fermented foods. In the starter cultures, the live microorganisms are added into this, into the raw materials, for example milk, and then we can produce the fermented products, curd or buttermilk, and so on. And the fermentation of sugars like by lactic acid bacteria, which produce lactic acid and other beneficial compounds. So this is commonly used for starter cultures in the food fermentation. So cheese is one of the most best example for the starter culture by using lactobacillus lactis microbes, and for yogurt, lactobacillus delbrueckii is used for the production of yogurt. So this is the short flow chart for the production of cheese manufacturing. So after getting raw material like fresh cow milk, we have to add the starter culture like lactic acid bacteria. After that, we can add the rennet for the coagulation process, and then next is whey separation. So we have to separate the whey. After that, we have to salting and stirring process for improving the flavor and moisture control. And next is pressing. For that, we can shaping the cheese and forming the finally we can aging for the maturation and flavor development. So next is application of lactic acid bacteria in adjunct and bioprotective culture. So adjunct culture which is nothing but the adding the additional bacteria or yeast or mold along with the starter cultures, and it do not produce the acid. It can improve the ripening and texture and flavor of the product. And in bioprotective cultures, so it helps to inhibit the pathogens and prevent the spoilage of the product. So the bioprotective culture also added into this for the cheese making and dairy industry and canned products. And next is the application of lactic acid bacteria in probiotic cultures. So in this lactic acid bacteria, which are diverse and wide group of probiotic bacteria, which is helpful in the gut microflora, which is faster colonization, and it also benefit to immune and metabolic health. So in this probiotics are microbial cell, which helps to prepare preparation and components of microbial cells, and this is very beneficial effect for the health and well-being. So some of the probiotic lactic acid bacteria are Lactobacillus casei, Lactobacillus ramonococcus, then Lactobacillus plantarum, then Bifidobacterium. So next, application of lactic acid bacteria in fermented dairy products. So this is nowadays currently using this fermented products. So first is cultured butter, and in this cultured butter, lactic acid bacteria species like Leuconostoc, Lactococcus cremoris is used for the production of butter. And then yogurt, lactic acid bacteria like Lactobacillus delbrueckii, then Bulgaricus, Streptococcus thermophilus, then Bifidobacterium are used for the production of yogurt. And kefir, so it is the a product. Kefir is a grain. The fermented milk product is excreted, that is produced by using lactic acid bacteria, Leuconostoc and casei. Application of lactic acid bacteria, this is a natural ingredient because this lactic acid is not only used for the fermented products, it also used as a preservative, it also used as an antioxidant, also enzymes and flavor component, it also used as a flavor compounds. And also this lactic acid bacteria which prone health and nutritional contributions because it is a probiotic culture which helps improve our digestive system, it control the intestinal pathogens, it also stimulate our immune system, and also reduce the cholesterol level, and it inhibit the tumors, and also it enhance the protein bioavailability, reduce carcinogenic risk, and finally it also helps to synthesis of B complex vitamins. So and finally, there is lactic acid bacteria which involve the new product development, particularly in cheese products. Nowadays, there are many cheese varieties are available. So low and reduced fat cheese, high flavor cheese, full fat cheese, and enzyme modified cheese. So all these cheese production, the lactic acid bacteria plays a vital role. And finally, this also lactic acid bacteria is not it's a good for health, especially elderly and baby foods. It helps to immune nutritive value and easily digestible, and the cereal carrying lactic acid bacteria cultures. It also used for foods, emergency foods, and in ice cream making. So the takeaway of this title lactic acid bacteria is, so it is a fermented products. It is driven in fermentation, safety, and flavor across the dairy product, not only dairy industry, also the meat, bakery, and beverage industries. So it helps to produce the enzymes, bacteriocins, then probiotic strains. It is nowadays used for beyond the traditional fermentation into the functional foods and nutraceuticals. And the ongoing enzyme innovation also implemented in this production of lactic acid bacteria. Thank you.